Hello again. So we're still talking about anatomical evidence, and we're going to continue with comparative embryology, where we're looking at how the embryo forms and the structures that form within the embryo until the organism is fully developed. And so if you look, you can see that different species, different groups have different structures that are similar. Invertebrates, all vertebrates have a pharyngeal pouch. So what this pharyngeal pouch is, and I'm going to show you, you have it right here. This is what develops into the fins, or not the fins, I'm sorry, into the gills of a fish and into the throat, ear and throat of a human. So we both have those. We also have the tail, which we've talked about. So we have a tail that eventually becomes a very tiny structure just called a coccyx. And a chick also has this, as do fish. Here you can see all the structures. We all start out with that little tail. We have those pouches. We have the brain, the same type of nervous system structures forming. But each of these are going to develop slightly differently, and eventually we're going to form a human or a chicken or a tortoise, salamander, or fish. The last type of evidence that we like to talk about is biochemical evidence. So biochemical evidence is very new evidence that we've only been looking at closely for the last few hundred years, or not hundred, oh my God, last few tens of years. So biochemical evidence is the DNA and ATP and the proteins that we can look at within different organisms. So when we look at on here a protein called cytochrome C, which is found in different animals that use aerobic respiration for energy, we see that the number of amino acid differences between a human and a monkey is almost nil. There's maybe one difference. Whereas between a pig and a human, there's, about close to 15 differences. A duck, there's even more. Turtle, fish, a moth, there's, almost, a little over 30 differences. And then we always have some outlier, something that yeast are not related to animals. We don't fall on the same line. So this is our outlier here. But it's also a eukaryotic organism, so there was a common ancestor between yeast and humans. So now that we know that evolution occurs, and I've showed you some proof that evolution does occur and that we can see organisms are very closely related depending on the type of organism. Now we're going to look at how we can see or identify evolution occurring. So for this to occur, we have to have some definitions. A population, in a nutshell, is just the members of a species occupying a specific space. The gene pool, then, is the total number of alleles within the population. So a population is considered the smallest biological unit able to evolve. You don't have an individual evolve. You're not going to just give birth to a new organism. That would be really weird. But what happens is within populations, as the genes change, evolution can occur. So microevolution is the change in gene frequencies that will eventually or can eventually lead to evolution. Macroevolution, where there's new organisms that evolve. I always like to talk about in microevolution. I don't have the picture, I apologize. Let me see, I know I have the picture. Oh, I'm just gonna go up one more slide so I can show you this. Here's an example of microevolution. So these are moths, and there are two different groups or types of the same moth. So there's the dark pepper moth, and then there's these light pepper moths. These moths have two different types of alleles. So if you have the dominant allele, then you're going to be a dark color. If you have two recessive alleles, you have the light color. And depending on the environment that these moths are found in, the darker moths may be more prone to being picked off than the lighter moths, or the lighter moths may be more prone to being picked off. So what what has occurred in the past is, prior to the what was it called? The Industrial Revolution, the environment was relatively clean. We didn't have a lot of smoke all over, and the dark pepper moths would sit on the bark of a tree and with the light pepper moths, but the bark of the tree was also a light color, and so the dark pepper moths were more able to be seen, and so they could be picked off easier. This led to a movement of gene frequency to the light color. But once the Industrial Revolution occurred, the soot in the air started falling, and it would start getting onto those the trees' trunks, and they become darker in color because of that. And that darker color allowed the lighter moths to be seen better. And so then we had that movement of allele frequencies back towards the dark moths. That's an example of microevolution. So now let's look at population genetics. Hardy and Weinberg are two scientists that came up with the same process known as the Hardy and Weinberg principle. And they used a binomial equation to help us identify allele frequencies and if evolution was actually occurring. So hopefully you remember that capital letters tend to be associated with dominant alleles. Lowercase letters are associated with the recessive alleles. And so the actual equilibrium equation is p squared. P squared is the dominant genotype, so the number of dominant individuals, plus 2pq, so that is the number of heterozygous genotypes in a population, plus q squared, which is the number of recessive genotypes in a population. And that would equal to 100% of the population for any given gene, which makes sense because all the dominants for one gene plus all the recessive plus all the heterozygotes would equal 100%. So if we're looking at this genotype frequency here, 0.04 is the number of big D, 0.32 is the number of big D, little d, and 0.64 is the number of little d. We could then figure out the alleles for each type of gamete. So big D has about 20%, little d has about 80%. And if we put those in place for these numbers, big D 0.2, little d 0.8, big D 0.2, little d 0.8. Take 2 times 2 is 4, but since it's a point, it'd be 0.04. Then you'd have 2 times 8, which is 0.16, but you have to add it to this 2 times 8 again. So you'd have 0.16, and then you'd take 0.8 times 0.8, which is 0.64. Add those numbers together, you'd have 0.04 plus 0.32 plus 0.64. So This is math. Some of you love math. Hopefully, you all will be able to figure out some of these problems. I don't ever ask really hard problems, but I do expect that you can figure out a basic Hardy and Weinberg problem. Okay? I will help you in class and in lab, especially if you're talking and asking questions, because that's what I'm here for. So let's look at what Hardy and Weinberg actually determined as the rules that will keep evolution from occurring. So what they basically said was that all frequencies within a gene pool will remain the same in every single generation of a large sexually reproducing population, as long as there are no mutations. So a mutation is a genetic change, a change on the genetic level of an organism. An example of a mutation that most people have probably heard of is a mutation in the insulin gene that causes, or in the, not insulin gene, in the sickle cell gene that causes, or I said sickle cell, in the hemoglobin gene that causes hemoglobin to form sickle shape, causing sickle cell disease. You can't have genetic drift. Genetic drift occurs when some environmental or some type of disaster occurs that causes the population to decrease greatly. Another type of environmental drift would occur if you had a small group of individuals that leaves a larger population and goes off on their own. Gene flow. You can't have gene flow, so you can't have populations coming in and out. So you can't have large groups of organisms leaving a population or coming into a population, because that would change the genes within the population. You have to have random mating, which means nobody gets to choose. It's random. Everything would be random. And there's no selection. So no organisms, no individuals are going to be more likely to survive than any other organisms. So we already know that none of these actually occurs. We know that there are mutations. We know that genetic drift can occur because when you have a natural disaster that wipes out a huge group of organisms, that's going to change the genes within that population. We do have gene flow. The U.S. is a great example of gene flow, as everyone comes to the U.S. But everyone has come to the U.S. To settle. So we are like that melting pot, if you remember from, I think, history. That is where we learned that. We have individuals from all other countries that come to the U.S. So there is gene flow. We have random mating, or we have random mating. No, maybe that could occur, but we typically choose our mates. We're very choosy, not just us, not just humans, but all organisms tend to be choosy. We want to mate only with the best, because depending on the organism, we may only produce a few offspring, and we want those offspring to be most likely to survive and reproduce on their own to make us very fit. And then there is selection. Different organisms are going to be more likely to survive in the same environment. So if you have two types, I'll just use the moths again, two types of moths, and one is easier to see, they're going to get picked off by the birds a lot easier than the other moths. That would be a selection. But what we can use, because we know how evolution won't occur, then we can use the Hardy and Weinberg equilibrium to determine if evolution has occurred. So here's another video. I'm not going to play it. Like I said, stop the video and watch it, and then you can actually understand a little bit more about the peppered moth experiment that they've done to see evolution in a nutshell. I'm going to stop here because it's been 15 minutes and I like to keep my videos relatively short so that you can watch and then be done and then watch the next one. So I will start up on the next slide talking about actual evolution. Okay.